Hello everybody, my name is Liu Zhongxi and my advisor is Professor Bingyue Xin. Today I'm going to talk about this article, Switching of Photocatalytic Tyrosine or Histidine Labeling and Application to Photocatalytic Proximity Labeling. First, let's introduce the corresponding author. His name is Zuoteng Senyi, and he got his PhD at Tokyo University. And his research is about molecular cell biology.
What is this article about? The proximity labeling is a method to know if multiple proteins have interaction or not. And these are some ways that people usually use to learn the protein and protein interaction. Co-IP, Y2H, FRET, and proximity labeling, PL. And here is the process of PL. The strategy of PL is to use a ligase which can link on the protein we want to learn, and this ligase can be activated by a specific trigger. Once it is being activated, the ligase catalyzes the covalent attachment of biotin to nearby proteins. When the biotin is attached to the proteins, we can use streptavidin to catch these proteins and then analyzed with spectrum to know what the protein it is. And this is an example. First, put BRA mutation to the objective protein and then trigger it. BRA can form a covalent bond between biotin and lysine in proteins. And the effective range is 1 to 10 nanometers. If the range is larger than effective range, then the biotin won't label on the protein. We can see in this figure, the black protein, biotin, are not labeled on the protein, but the red one are labeled on some protein which is nearby the BRA mutation and also the objective proteins.
Then let's talk about this article. This method, the photocatalytic proximity labeling, is using the photocatalytic in the proximity labeling method. And it has more precise effective range than the previous one. The range is about 6 nanometers. First, the lab made this compound called MAUI, which can label on the protein with the help of photocatalytic. There are two different reactions to label on the protein, which is the single electron transfer pathway, which can label on the tyrosine, and the singlet oxygen pathway, which can label on the histidine.
Because there are two different mechanisms, the author wants to know what role does the photocatalytic plays in it. And here he chose 15 different photocatalytic, and he wants to compare which pathway does this photocatalytic works. So by using ubiquitin, which has only one histidine and one tyrosine, and put 15 different photocatalytic. After lysis with trypsin, and we can compare the number of tyrosine and histidine to know which pathway does these different photocatalytic works. And we can see the left figure. The blue one is the tyrosine labeling, and the red one is histidine. There are three different compounds are very special, which is the number 2, number 7, and number 15. The number 2 has almost the same histidine and tyrosine, and the number 7 has a lot of histidine than the tyrosine, and the 15 has even more tyrosine label.
For a double check, the author want to check if these compounds are actually labeled because of the single electron transfer and the single oxygen pathway. The left figure is to track the fluorescence intensity of the compound 15, which is using the single electron transfer. When the single electron transfer happened, the compound won't have the fluorescence intensity. So by increasing the MAURA, the intensity of the compound 15 will decrease. Like when it adds 0 millimolar, the intensity is bigger than when it adds more, like the 100 millimolar. In the right figure, at first, this is the production of the single oxygen. At first, the production are all the same. And after being using the single oxygen pathway, the production is being decreased. And compound 7 use it more than others. After irradiation, the fluorescence intensity of compound 15 decreased a lot. Then the compound 2 and compound 7, the photo stability is better than 15.
Then Arthur tried to know if this method can really use to check protein and protein interaction, so he designed this experiment. Halotech and its ligand are known to bind together as a protein and protein interaction. First, Arthur linked the photocatalytic to the ligand and put all these things in cell lysate. In the cell lysate, the halotech and its ligand will bind together, and so does the photocatalytic. If the halotech is near enough to the photocatalytic, the effective range, then the MAURA will label on the halotech. And at this time, the MAURA is linked with azide, which can be recognized with the Cy5. And then put all these things into SDS page. If the result show that we can catch the molecular weight of this halotech and the Cy5 signal only after irradiation, then this work is success. And let's see the result. First, at the right figure, we can see that the halotech's molecular weight is around 55 to 70 kilodalton. And the bar 2 to bar 8 are all the cell lysate, which has a lot of unknown proteins. And we can see this result. This is the Cy5 signal. We can only see the result. We can only see the Cy5 signal after light irradiation. And we can see the halotech's molecular weight are very, very clear. So I think this result shows that this method can really recognize the protein and protein interaction. And this is a Holotech H2B photocatalytic proximity labeling in cell. And the other only changed the azide to biotin. And using this dye, the strep to the Texas red, and strep can bind with the biotin. So these results show that the photocatalytic proximity labeling can work in cells. Thanks for your attention.